Applications of PCR. Now we have discussed different variations of the basic PCR reaction. Although while discussing different type of PCR, briefly we also discuss its application, that for what purpose it has been developed and why it can be used. Now PCR, it has been widely used in the discipline of life sciences, either it is a field of molecular biology, genetic engineering, or any other discipline that is of biological sciences. Now, one of the technique that we are going to discuss is that it can be used in the study of gene cloning and expression. If you remember, while discussing cloning strategies, we discussed that how PCR it can be used to screen different genomic libraries or in the cloning procedure. What is the role of PCR? Normally, while isolating or screening of our gene of interest, normally we extract the DNA and it can be digested with restriction enzyme, and after digestion it can be ligated with a particular carrier molecule, that is the vector, so that we can produce recombinant molecules, and then it can be inserted into the target organism. That is, transformation step is there, and after transformation the laborious process of screening it can be started. So in normal process of genetic engineering, where whole genome is involved, so different recombinant molecules can be produced that also contain our single gene of interest among thousands molecule. So in this way we are going to produce gene or genomic libraries. These gene and genomic libraries, they are the representative of whole genome, and we are going to isolate our gene of interest. So it is very time consuming process, laborious process, need lot of expertise. But this procedure, it can be made more selective when we are going to use PCR. PCR यूज करने के लिए ज़रूरी है कि हमारे पास जीन से रिलेटेड स्पेसिफिक प्राइमर्स भी हों. इसका मतलब है कि अगर हमने किसी जीन को टारगेट किया है तो उसका हमें नॉलेज होना चाहिए कि उसका सीक्वेंस क्या है, ताकि हम उससे जो स्पेसिफिक प्राइमर हैं वो डिज़ाइन कर सकें. अगर तो हमारे पास ये नॉलेज है तो जो फर्स्ट स्टेप हम यूज़ करते हैं कि द कंस्ट्रक्शन ऑफ जीनोमिक लाइब्रेरीज, उसको अवॉइड किया जा सकता है. इसमें हम प्राइमर कंस्ट्रक्शन के बाद हम स्पेसिफिक एम्पलीफाई करते हैं जो हमारा टारगेट जीन होता है, ताकि उसकी बहुत सारे कॉपीज प्रोड्यूस हों. देन आफ्टर एम्पलीफिकेशन अ स्पेसिफिक जीन इट कैन बी ligated into the vector, and then transformation it can be induced. So it is relatively simple process if we are going to use PCR, so that we can get our target DNA amplified by using this technique. So here PCR, it is also playing its role in gene cloning. On the other hand, once the organism has been transformed, then different screening strategies it can be employed. Like if we have used the genomic DNA insert that has been amplified by the PCR, and even we discussed that messenger RNA it can be converted into cDNA, so that the cDNA cloning it is there. And again the messenger RNA it can be amplified by using PCR. So cDNA it can be ligated into the vector, so that it can result the cDNA libraries or genomic DNA libraries. So both of these libraries, once they are formed, they can again be screened by using PCR. Traditionally, How we are going to screen the cDNA libraries or genomic libraries, that sequencing technique it can be used, कि हमने जो जीन इंसर्ट किया है उसको आइसोलेट करें, उसका सीक्वेंस करके देखें कि क्या ये हमारे टारगेट जीन से रिलेट करता है या नहीं करता. तो सीक्वेंसिंग इट कैन बी performed, and sometime the probe can be used by the process of hybridization to detect the target insert of the DNA or a particular gene. In addition to that, in order to detect the protein product, immunological technique can be used. But again here, the PCR, it is also playing a role in the screening strategies, that whether a particular organism it has been transformed or not. So here the DNA from the transformed organism, it can be isolated, and then specific amplification it can be used by using some specific primer with the help of PCR technique. So PCR, again, it is showing that a particular target gene it is present in that host organism or not. So PCR, it has been extensively used in the cloning of the genomic DNA or the complementary or cDNA, and in addition to that, it can also be used in the screening of the genomic or cDNA libraries. So it also finds its application in the field of gene cloning or genetic engineering.